In today's presentation, we're going to be focusing on how to build up excellence in management of clinical trial site reads, and it's titled Do Not Resist RECIST 1.1 and All Other Criteria.
Thank you very much, AJ, for the introduction. I also would like to warmly welcome you. I'm Tobias, and I would like to start over in presenting the big picture, the landscape for imaging in cancer research with all involved stakeholders. This is a quite full slide, but let me guide you through. On the very right side, we do have the authorities, FDA and EMA, deciding about cancer drug approvals. Next to it, we have the pharma companies who set up clinical trials to get approvals for developed drugs. The imaging CROs, or CROs in general, operationally support the clinical trials as a service provider for pharma, and they generally do the so-called central read workflow. For example, with double-blinded reads for images, of course, depending on the trial protocol. But of course, all cancer patients who participate in clinical trials are treated in hospitals. And there you can differentiate the job of a radiologist as follows. First, every cancer patient in clinical routine gets a clinical routine read, independent if they are participants of a clinical trial or non-participants of a clinical trial. But second, when a patient participates in a clinical trial, they normally get a dedicated clinical trial site read, where specifics of a trial have to be considered, like specifics of criteria. And last but not least, third, radiologists also work on their own research projects for example, doing their publications. Therefore, they also have to do research reads. Lesion now, our software platform, we are providing is currently used by different stakeholders and is designed for different jobs to be done, like in pharma for imaging biomarker research or criteria research, like for CROs to conduct the central read workflow, but also at sites where we support all three use cases. Important for you to understand that we do not develop dedicated software solutions for each of the different stakeholders. It's just one software we are developing, and we just configure it in different ways. The main focus of today's webinar is on clinical trial site reads. But if you have a major interest in the other areas, please let us know, and we will follow up after this webinar. Before we now do the deep dive, I just want to show you the result of the question we raised when you registered for this webinar. We ask you how many clinical trial site reads does your department do per week? We had 123 valid answers, and they were split as follows. 42% said 5 to 10 reads per week, 16% said 10 to 15 reads per week, 10% said 15 to 20 reads per week, and almost 1/3 of you are doing more than 20 clinical trial site reads per week. For us, it would be quite interesting to learn how you currently manage those reads, and we are very much looking forward to discussing this with you after this webinar. Let us now specifically focus on the clinical trial site read, and first we would like to present you two different scenarios, how those site reads are organized. First, some institutions perform their site reads in a so-called primary read workflow, meaning they just do their routine read, and within this process, they also document all requirements for the clinical trial assessment within an Excel-based tumor log or even with paper and pen and dedicated forms. Another scenario is to do the site read in a dedicated secondary read workflow independently from primary routine read. Actually, the majority of our sites we are in contact with worldwide apply this secondary read workflow. I think there are pros and cons for both scenarios, but I believe the most important argument for a secondary read workflow is the expertise that is required by the radiologist who performs those site reads. On the following pages, we will now focus on the secondary read workflow, but we also have customers using the primary read workflow, and they're very successful in doing it. So if you are very interested in how to perform clinical trial site reads in primary read workflow, also let us know, and we will give you some more information on this. Let us now have a look into a specific site doing this clinical trial site read in a secondary read workflow, And let us first introduce the different stakeholders who are involved. We have, on the one hand, a sponsor representative who has to ensure that all clinical relevant information will be captured and documented in a clinical trial charter conform way. We do have the principal investigator in oncology who has the overall responsibility of a trial in a site, and generally there are clinical trial coordinators to support the PI. There are also clinical trial coordinators in radiology supporting the clinical trial work in this department, together with a technician who ensures that functionality of all systems that are required. But of course, last but not least, there's the senior radiologist who is doing the clinical trial site read. Without a dedicated system, they normally work with 3 different instruments. First, they have the PACS viewer to view the images and to measure the lesion. Second, there are some forms for the documentation of the results, like Excel-based tumor logs or paper and pen-based forms to fill out. And last but not least, they have also to consider the criteria with all complex details that might come up. Before we now show you a workflow or how Mint Lesion helps you to manage this workflow, let us have a short look into the criteria world. What are criteria? Criteria are a kind of rule set to apply in therapy response assessment, leading to a dedicated time point response. For example, saying the cancer disease is stable, regressing, or progressing. Why do we have different criteria? Different cancer types and different therapy approaches require specific rule sets to assess a cancer therapy. One example are the upcoming immunotherapies, which have a total different mode of action compared with standard chemotherapies. Radiological response assessment has to be aligned with those different mode of actions, and this is why more and more criteria pop up. When we now consider the example of RECIST 1.1, there are some image-based input you have to capture, quantitatively as long or short axis, but also qualitatively like the documentation of non-targets or the appearance of new lesions. Together with a certain rule set that is defined in the RECIST publication, time point responses are derived to describe the radiological perspective of the cancer therapy response assessment. This criteria aspect is one of the most complex one when doing a clinical trial site read. Let's now have a look how Mint is supporting this workflow, and for this I hand over to AJ.
Thanks, Tobias. So as you were mentioning, what we generally find with our current in the market right now is the use of 3 non-integrated instruments: the PACS viewer, a paper-based tumor log form, and of course, a publication or the trial charter that has to be followed. But with Mint Lesion, we combine these 3 elements into one intelligent, cohesive platform, which first of all is an image viewer in itself. Secondly, it knows all the criteria. So Mint knows RECIST 1.1, iRECIST, Lugano, Choi. All of these criteria are built into the system, and it will guide the radiologist with the assessment that they have to make, prompting them with questions that they need to answer. For example, perhaps where they have to classify the lesions that are measured, simultaneously ensuring that the conformity of the criteria is being met, and finally, documenting all these results in a standardized and structured way and generating reports. So to demonstrate this, let me just launch the application really quickly. So the first step is basically to set up the study. So it depends on the workflow. You might have a clinical trial coordinator who is going to enroll the patients, model the trials, and assign that to a certain radiologist. So let's take a look at how this would be done within Mint. So first, the coordinator will go into the application and click on Add Trial. Now they have the ability to give the trial name. In this case, let's just say webinar demo. Now you can add multiple trial arms within each criteria or within each trial. Let's say there are 3 or 4. You can also easily remove them by clicking on this minus button. So the application is very intuitive. Suppose there are more than one criteria that needs to be used. So each trial arm has its own separate criteria. That's also possible. Let's say we have RECIST and iRECIST and maybe Lugano. It's very easy to select this and just quickly modify the trial to the way you want. You can also assign default users or assign the cases to either users or groups of users. And in this way, you have the possibility to let Mint coordinate all the communication of notifying if there's a new assessment that needs to be done and automatically sending the PDF. You always also have the ability to add some more information in the description box. So in this way, now I have just set up a very simple trial with RECIST 1.1. Next, I need to enroll patients into this trial. So I can go through the patient list to identify the patients I need, or I can go to the DICOM query button to look directly in my packs. Once I have my patient, I can just search for them with an ID or a name. And then when I click on my patient, I can see the timeline of all the different studies and cases that are relevant for this patient. So here I can see that, for example, this patient has had a first-line therapy, which was progressive. Then this patient is on another trial. Let's redo this trial, and I'll show you how easy it is. So first, I'm going to add patient to the new trial that I want, clicking on this button up here. Then I select the demo trial that I've just created. Now I have a case which encompasses the entire time, all the time points. But let's say I want to skip the first two. I just use the minus button, and this way I can adapt the case to the baseline that I need. Now I have my baseline, and I can get ready to perform my assessment. So now, when the radiologist comes into the application, he sees something that's quite familiar. For example, you have your different series on the right-hand side that you can just drag and drop really easily. The application behaves like a PACS viewer. You can scroll, you can change level window, zoom, pan, and just reset everything with a simple click of a button. But what's different is on the left-hand side. This is where we have the smart bar. So you can easily access, for example, the main information that is relevant for this case, RECIST 1.1, and see things like, okay, there's a maximum number of 5 target lesions that I can have and 2 per organ. So it's quite easy to just see the main important points. Let's say I have identified this to be my target lesion. I go to the target lesion category and click on the plus button. Now Mint will prompt a tool, which is the minimum requirement, which is just a ruler. But since this has a nice clear boundary, I'm going to use this really cool tool, the semi-automated volumetric tool. With this tool, I just have to click in the middle and define a rough threshold for the lesion. Then Mint will automatically segment this lesion in 3D and take the slice which has the largest diameter. Not only that, this tool is also capturing, for example, the volume, the density, and it's also capturing texture parameters in radiomics. I can even go and set the different threshold and filter values. But you can see that up here now, Mint is giving me this error. It's telling me that, hey, you haven't defined an organ location for a target lesion. So, okay, I'm going to go back and click on liver, segment 8. Now that conformity check disappears. So let's go ahead and document another one, another target lesion. There's another lesion down here. So I'm going to click on plus for target lesion. Mint gives me the main tool, but I still like this tool because it's a lot more easier. And I capture so much information that without any extra effort, and I can always go back and use all this extra information for any kind of study that I want later on. Mint will automatically take the right value for the long axis and is capturing all of this extra texture analysis and details. I classify the organ location and I'm done. So I know what you're thinking. AJ, can I use this tool for lesions like this? There's a little bit messy. And the truth is, well, it's probably not going to work in the way we just saw, but we have a different tool for this. So I'm going to use this ROI tool and just drop some control points around the boundary of this lesion. And in this way, I am just encompassing the entire lesion and letting Mint calculate the right long axis. So there's no longer the error of one radiologist eyeballing the long axis in a different way. But here I still am capturing the area, the density, and texture. I'm going to say liver segment 8 again. And now if I classify this as a target lesion, Mint is giving me another error that I am putting more than 2 target lesions in the same organ, which is not allowed. So in this way, we're continuously checking the conformity. I'm just going to drag and drop one of these into non-targets. And always there is the scope for the radiologist to add some more information. And even though Mint is a very structured application, we still provide a lot of flexibility and customization. So in this way, I have very easily conducted my assessment for baseline, and I can hit approve, and Mint will automatically send this report, generate a report first of all, and send it as an email attachment to my study team. But the fun really starts when I have to do a follow-up assessment. So in a follow-up assessment, Mint, first of all, can auto-retrieve the subsequent time point studies for the patient on a trial automatically and notify the radiologist. Then when the radiologist clicks on the case, he's brought to a screen where he can see the current time point compared with the previous time point for each lesion. Mint will bring the lesion and show you how it has progressed in the timeline in the bottom, and bring you to where the lesion should be in the current time point. It gives you the same tool that you used last time and will automatically segment the lesion for you. Again, it's capturing all the relevant information. Similarly, I can jump to my next lesion, let Mint synchronize the slices for me and give me the tool. I can make a little bit of a modification. I can change the tool so that flexibility I mentioned is always there. Same step, Mint will synchronize. I use the smart tool. And in this way, I have now captured 3 lesions over all of these time points in a matter of seconds. When I go to the report area, which is the third and final area in the Mint application, I can see the wealth of information that I have now generated with very little effort using the smart intelligent assistance that Mint provides. Mint is now generating a lot of results and report segments, like, for example, a text block, which is a comprehensive analysis. But at the same time, it provides a lot of graphical information and graphical details. So, for example, a probability density chart, histogram, stacked area chart, and even images, so the snapshots of each lesion that was measured, along with the slice and series that it was measured on, the modality. All of these images are scaled and can be exported as you wish. It generates tables for each target lesion, non-target lesion. These tables can also include the images themselves, and there's always the scope, like I mentioned, to add some more information or comments or summary details at the various different text blocks. Now, finally, let's go to the report. So when I hit on approve, Mint is going to generate this report for me. And let's take a closer look. So here's, for example, the comments that I added for the summary, some patient information, trial information. The first page has the table where I can see, for example, the time point response that Mint has calculated. The second page has a stacked area chart, which is useful to analyze mixed responses. Very easy to see this. As we scroll further down, there's also a target sum chart. This line helps me understand what the region of progressive disease, stable disease, and regressive disease are. And it's very easy to understand, okay, if the patient has a trend which is getting better or worse, and helps make a therapy decision as well. Towards the bottom, now we have the snapshots of each lesion, like I mentioned, scaled, along with, the change to the previous time point. We can have the slice and series. And finally, a conformity check page where we can see if any of the conformity checks were not passed or any revisions were made to the report or the assessment as well. So in this way, we can really see how Mint transforms your workflow, and in this way enables you to really be empowered to produce high quality reports in very short time.
Thank you very much, AJ, for this fabulous presentation and showcase. What did now change when we apply now Mint Lesion to the secondary read workflow? Actually, what changed, every stakeholder that is involved has now a smile in his face because they are very happy, on the one hand, doing the read with Mint Lesion, but also receiving the results from Mint Lesion. And also the sponsor representative can really reproduce all results he gets from radiology regarding a clinical trial site reach. Just to give you another information regarding how secondary read workflows could be organized. In a lot of our institutions who apply Mint Lesion, also this person in here, the technician in radiology, takes over a very important role. In those institutions, the senior radiologist is preparing the baseline, but technicians taking over the task of doing and preparing the follow-up assessments. So the senior radiologist only jumps in to validate the reads technicians did, and so they minimize the time they spend for clinical trial site reads. And this is also possible in Lesion because you can every time reproduce what was prepared for you. Actually, there's only one thing left to say. It's thank you very much for your participation.